The broadcast is now starting. All attendees are in listen-only mode.
Hello everyone, and thank you for joining us today. You've logged on to the Nelson Labs webinar, Disinfectant Efficacy Testing, Study Design, and Data Interpretation for the U.S. Environmental Protection Agency. Now, if you missed one of our webinars, or like to simply refer back to one, you can always find them on the Nelson Labs website on the On-Demand Webinars page listed under Education. You can receive notifications of upcoming live and on-demand webinars and seminars by liking Nelson Labs on Facebook or following on Twitter or LinkedIn. We also invite you to register for free for the Sotera Health Academy at soterahealth.com/academy. There you will find curated content from industry thought leaders, from introductory sterilization and lab testing topics to advanced in-depth learnings. Our expert advisors have filled this academy with cutting-edge educational content and resources to help you mitigate risk, go to market faster, and achieve excellence in your field. Nelson Labs is a leading global provider of laboratory testing and expert advisory services for medtech and pharmaceutical companies. The company performs over 800 rigorous microbiological and analytical tests across the medical device, pharmaceutical, and protective barrier industries. The experts at Nelson know every test matters and requires solutions to complex problems to improve patient outcomes and minimize risk. So let's get started. Today we're joined by Thalia Contreras and Grady Wertman. Thalia has worked at Nelson Laboratories for over 2 years. Initially, she started as a laboratory analyst in November 2019 within the product sterility department. Through her eagerness and ability to learn quickly, she transitioned to a floating study director position in May 2021. This has allowed Thalia to work and expand her knowledge base within microbiology, chemistry, and molecular testing of pharmaceutical and medical products. Grady has worked at Bioscience Laboratories, a division of Nelson Laboratories, for over 8 years. He started as a laboratory technician in December 2013 without any lab experience, but since that time, he has become a study director 3 at Nelson Labs and has made it his goal to learn and deeply understand any and all methods utilized. Now, beginning today's presentation, I'd like to turn the time over to Thalia and Grady.
Thank you, Mike. So today we are going to be discussing how to choose the appropriate methodology and how to best support a label claim for submission to the United States Environmental Protection Agency. So in order to substantiate the claim the sponsor is going to make on their label, they must perform one or more evaluations as dictated by the EPA guidance.
The overall goal of this presentation is to give you a general overview of disinfectant efficacy testing and to provide you with some detail so you can choose the best methodology to assist you in designing studies for submission. As we go over the testing, methodologies, and strategies, we're going to go over the test method acceptance criteria and interpretation of these studies. Specifically, we aim to provide insight on how the results of a disinfectant efficacy evaluation is gathered, interpreted, and best presented for sponsor submission. To begin, we just wanted to go over some general definitions per EPA. A sanitizer is something that is considered a chemical surface agent that kills bacteria. Currently, it is not intended to kill a virus. However, with ever-changing science, EPA is in discussion of potentially accepting claims for sanitizers against viruses. Currently, disinfectants are known as chemicals for surfaces to kill viruses and bacteria. A sterilant is the same as a disinfectant, but includes a sporicidal claim. And just as a note, EPA does not regulate hand sanitizers. This falls under the purview of FDA. EPA focuses on environmental surfaces, whereas FDA focuses on body surfaces. Along with these definitions, I just wanted to point out some key terms that you might hear as we go through the presentations. This is a carrier, which is something that is infected or is carrying that key microorganism that we're testing against. In testing, this can be either a steel penny cylinder, a porcelain penny cylinder, a microsoap slide, or a silk tube. It varies depending on the test method required. The test system is interchangeable to being known as the test organism, or sometimes called the pest, which is the challenge microorganism that we're going to be testing against, or is listed on the label claim. A label claim is the actual what the product is saying it can do, what it's going to be killing against, and within what time frame. That is what the label claim is. And something that is RTU is ready to use. For example, is something a disinfectant or sterilant that doesn't need additional dilution to actually be put into practical use. I provided here a list of the general chapters within EPA. Each disinfectant or sterilant will fall into one of these general chapters, and the chapters themselves have further detail into specific test types. There's various methods to testing disinfectants, such as carrier tests, capacity tests, practical tests, field tests, and even in-use tests, each with its own disadvantages and advantages. The goal is to choose the best method for your disinfectant or sterilant that is replicable to a real-time use example. As we go through, you'll be able to see how to use these EPA guidelines to choose the appropriate test for your sample. Each test will encompass, of course, the carrier type, the contact time, and also the best and worst case scenarios for that contact time. Now let's dive deep into some more information.
So getting into the 810-2000 is the general considerations. This guidance really goes over the key fundamentals that are needed for testing and for developing the label claim. So the critical testing requirements are that your product must be tested at the lower certified limit, which is the nominal concentration minus 3, 5, or 10%. Based upon the concentration of your active ingredient, which more can be determined from 40 CFR 158.350. So the purpose for testing at the lower certified limit, the level below your release concentration, is to show that the product works below that concentration, and so that when it's released on the market, the efficacy is there and not tested in lab at a higher than release concentration. The method used should be relevant to the pest or the test system, the use sites, and the claims that will be made on the label. As Talia had said, testing should reflect the patterns intended for the product. And if it's going to be used as a food contact in a kitchen, it needs to be tested under those conditions. If it's going to be used in a laboratory, there's different methods that need to be followed. A new method may be developed if no existing method is present, but that does require consultation with the EPA so they can approve it prior to just receiving the packet. The test organisms are chosen based on the projected label claim, but specific method microorganisms must be used to acquire the base label claim before additional species can be added to the label. Where dilution of the product is required, as it may be supplied as a concentrate, in testing in the laboratory, hard water must be used. This can vary from 200 ppm to 400 ppm calcium chloride to 375 ppm OECD hard water. It depends on where the use is designed, where the product is designed to be used. The contact time in testing should be the same or shorter than the contact time identified on the product label. And neutralization is needed to be proven that the test material is inactivated at the contact time. So the first guidance we're going to go over is the 810-2100. This is for sterilants, sporicides, and decontaminants. The key takeaways from this slide are just kind of to get familiar with the required testing for different formulations. So for the sterilant and sporicide, they are nearly identical with only minor changes between the two. So the liquid water, soluble powders, and gases are going to be tested the same way using the AOAC 966.04 between method 1 and 2. And then any sprays, mists, foams, gas vapor with use of device will require agency consultation prior to the beginning of this study. So for each of these two types of claims, you're testing the same two species, and each organism requires 3 batches of the test product at LCL, 60 carriers per batch. For a sterilant, you're required to test the porcelain penny cylinders, so a hard porous surface, and a silk suture loop, so a soft porous surface. Additionally, for the sterilant, verification of an additional batch at the LCL, but only using 30 carriers is required. However, for the sporicide, you're allowed a little bit more leeway on the surface. So for the sporicide, you can indicate testing is only for hard non-porous surfaces, so a stainless steel carrier or a similar carrier, or only for the porcelain penny cylinders or the silk suture loops. So that's a little bit there. And then if another very important one is testing for Clostridium difficile spores for use in hospital and healthcare disinfectant areas. So this doesn't necessarily supply a direct method to use, such as the AOAC method mentioned before, but it actually links to a website on the EPA's webpage that gives explanations and provides the SOPs that are to be used to develop the spores and the testing involved with those.
Going into farther detail with these methods, I just want to emphasize that these antimicrobial pesticides are intended to be used on inanimate, environmental, even food packaging surfaces. There are 2 methods that are mentioned. The first one focuses on Clostridium sporogenes, which, if you don't know, it's a gram-positive bacteria. If somebody is infected with it, typically it produces a type of toxic gas inside a human body, and it's very commonly found in packaged food where oxygen levels are low. Despite this, it is considered a level 1 organism by ATCC, which makes it perfect for laboratory use and replicability of the study. Bacillus subtilis is what method 2 focuses on, which is also a gram-positive bacteria. It's non-pathogenic. However, it has a high heat resistance, so it's often found in food poisoning, which again is why it makes it a perfect organism to use in testing. The focus of these test types is to concentrate the organism at a 1 × 10⁵ dilution through a 1 × 10⁶ dilution. And it should be tested at 3 batches of this test substance with 60 carriers each. What makes these methods so easy is that you can use it on any relevant carrier surface. So oftentimes Clostridium will be used with silk loops, which the method also specifies how to make them. And then Bacillus will have penny cylinders that can be either porcelain or stainless steel. And these carriers will be submerged into the liquid organism, then introduced to a disinfectant, and then a nutrient bath with a neutralizer. That's the general flow of how these tests will work. This test is heavily qualitative, so the success rate is based on how much growth is seen. To be completely accepted, it has to see no positives within all of the carriers, all 60 of them.
The next one is the testing for Clostridium difficile. So in order to test these, the spores for the Clostridium must be made in lab following the EPA standard operating procedure MB28, which describes the procedure for production and storage of the spores. And then for the testing, you're following the standard operating procedure MB31 for evaluation on an inanimate, hard, non-porous surface. So this study is conducted on small stainless steel penny cylinders, as seen in the pictures, where the spores are dried onto the penny cylinders and then exposed to the liquid test substance and neutralized. So this is a quantitative evaluation. So this one, all the recoveries are diluted and plated, and the reductions quantified based upon reductions compared to the control carriers. So for success for this one is a greater than or equal to 6 log 10 reduction when compared to those carriers. And also the spores must be qualified in testing using sodium hypochlorite at 1500 plus or minus 150 ppm as a test system control to verify that the spores are hardy enough to justify testing. This one is, so now we're moving on to the 810-2200, which is the disinfectants for use on environmental surfaces. This is one of the more important tables that a lot of people use, in that it offers guidance on the type of claim or label that is going to be sought after. So the first claim that can be acquired is a limited spectrum claim, which is either for gram-positive using Staphylococcus aureus or Salmonella enterica. So that's going to be the methods chosen for each of these 3 options are the same. The only thing that differs is which organisms are utilized for the claim. So the next claim would be a broad spectrum. So this one encapsulates Staphylococcus aureus and Salmonella enterica, and then Pseudomonas aeruginosa can be substituted for the Salmonella if necessary. And the third claim is for hospital or healthcare if the product is to be marketed to hospitals or healthcare facilities. The testing must be performed using Staphylococcus aureus and Pseudomonas aeruginosa are the required ones for that. Salmonella enterica is not allowed for that claim. So if the product is a water-soluble powder or liquid, the used dilution method will be utilized. If the product is intended to be shipped as a spray product, it'll be the germicidal spray product as disinfectants test method. And if it's a towelette, it's a slight modification from the spray disinfectant test to utilize those towelettes.
In continuing, these environmental surface disinfectants definitely vary widely. If they're tested against bacterial claims, they definitely need to meet that base bacterial claim with supported data. But if it's labeled otherwise with additional fungicidal, virucidal, or tuberculocidal claims, additional testing and evaluation is needed. I want to emphasize that these all need to meet that less than 10-minute contact time. It's very important for these types of disinfectants. Going into the first 2, the first focuses on internal toilet, urinal bowl kind of cleaning disinfectants. It's more of a niche area, but it's definitely something to focus on, I guess. If you see things like products like ZEP at home or in public restrooms, those are products that are typically tested under these. They're tested at a limited spectrum or a broad spectrum. So like I mentioned, sometimes you need to have a disinfectant meet a base fungicidal, virucidal, or tuberculocidal claim. So a broad spectrum is a claim that encompasses all 3 of the major classes of organism, which are bacteria, fungi, and virus, as mentioned. So below that are the virucidal claims, which follow ASTM E1053. These claims are, or these tests, this test method is heavily quantitative. So it really focuses on a log reduction type of study, comparing a log reduction from one product to the next. The calculations are usually between Reed and Munch, most probable number, or Spearman-Carber, getting that tissue culture lethal dose value. The below two are the ones I'm going to focus on the most, which are the fungicidal disinfectant and tuberculocidal disinfectant claims. I'll get into more detail with them as we go on in the presentation. But to summarize, they follow either a use dilution method or a germicidal spray test method with an appropriate organism to each. For fungicidals and tuberculocidals, they just consider 2 batches with 10 carriers per batch. And they can either be a water-soluble product, a spray product, which covers is under germicidal spray test, or a towelette, which is also under germicidal spray test with some modifications. So going into more detail about the disinfectants on environmental surfaces testing, like I've mentioned, these are for water solubles, powders, and liquids. They typically follow AOAC use dilution method. So as a general picture for use dilution method, it's a total of 60 carriers are typically used, which can be, again, as mentioned, the penny cylinders of porcelain or stainless steel. These penny cylinders are initially introduced to the test system by submersion for a total of 15 minutes or more, depending on the method written. And then it is taken and introduced to the disinfectant. The disinfectant can either be a diluted version of the disinfectant or a ready-to-use version, depending on the study at hand. It's introduced to the disinfectant for the particular contact time that is on the label claim. And then from there, it is taken and placed into a growth medium with neutralizer. Once it's in that growth medium with neutralizer, it tends to be incubated for a certain amount of time and observe growth within that time. This is really focuses on assessing rate errors, both a pass error and a fail error. A pass error is typically a disinfectant being tested at a low efficacy, but it's deemed pass because of the performance standard through the use dilution method. If it's a fail error, it's for disinfectants with acceptable efficacy, but it's usually failed based on the performance found within that use dilution method. Typically, for use dilution method, which I'll get more into later, it's 59 out of the 60 must not show growth through them. The other test method for spray products and towelettes are the germicidal spray test. First general picture, this is very similar to the use dilution, where the organism is going to be spread over, is going to be introduced to a carrier and then sprayed with a disinfectant and then transferred over to that liquid growth medium with neutralizer and observed for growth after that. If it's a towelette, it's typically folded in a special folding manner as the method will describe, and tested in that same similar manner. Introduction of an organism, then introduction to the disinfectant, and neutralized to ensure that the contact time is met exactly.
Okay, so one of the first methods that Talia had talked about was the use dilution method. So this method, like she said, is using stainless steel penny cylinders or porcelain penny cylinders. And it's a very qualitative evaluation in that nothing is plated for direct recovery. The passing and failing, the success of the product is based upon growth seen throughout the 60 carriers. So the requirements for this are to test Staphylococcus aureus, Salmonella enterica, and or Pseudomonas aeruginosa. 3 batches are required. 60 carriers are required to be tested per batch per species. For the use dilution method, each batch for Staph and Pseudomonas must be tested on a different day to eliminate the day effect that has been seen in the use dilution. So for the used solution specifically, the number of positive carriers that are allowed to be considered passing change for each species. However, they're very strict, and any additional positives result in a fail for that batch. For the germicidal sprays and towelettes, each species can only have one positive carrier and still be allowed for passing. So using these tests, you can test for a limited spectrum claim, a broad spectrum claim, and or a hospital healthcare disinfectant claim for hard, non-porous surfaces.
As mentioned, fungicidals and tuberculocidal claims are very similar to the previously discussed methods, but with minor modifications to really emphasize the base claim for fungicidal and tuberculocidal. And they are modified so that only 10 carriers are required and only 2 batches are required, but absolutely no positives are accepted in these methods. To focus in on fungicidal testing, they either go by AOAC fungicidal evaluation testing, which is AOAC 955.17, or AOAC use dilution method modified for fungicidals. The main focus organism are Trichophyton and Pterididitae. And looking at fungicidal activity of disinfectants, that type of testing is definitely a suspension test. And it fully evaluates the efficacy of the product. If it's a use dilution method, the test is typically modified to conform with the appropriate element. So either the media changed or the growth conditions are changed. And these are typically added to a broad spectrum type of testing to encompass all the requirements for the target organisms. They're also, these test methods are sometimes used for hospital types disinfectants. But of course, this is all subject to agency approval, since any modifications to the method needs to be discussed with EPA prior to use. For tuberculocidal testing, again, this is also used as a broad spectrum and can be hospital disinfectants as well. But it focuses on either glutaraldehyde-based product versus non-glutaraldehyde-based products. These are, if you are wondering what these are, they're so typically under brand name. So you'd find it under like something like Cidex or glutarol disinfectant. Typically, these, if it's with glutaraldehyde, it is used as a sterilizer for surgical instruments and other areas of the hospital as well. As a cool tidbit, it is also used for medication, typically to use against warts on the bottom of feet, if anyone's curious. But that's what this test method focuses on. Its main target test system or test organism is Mycoplasma bovis, which, if anyone is familiar with mycoplasmas, are a very strong organism to fight against. The benefit of this germicidal spray test is definitely that it's a high-level test for spray disinfectants, meaning it's very good at killing germs at a relatively short amount of time. But a weakness is that it's very fundamentally similar to that use dilution method. It's only a slight modification.
So focusing back on the used solution, I put together this base claim matrix to kind of help guide, the differences between what are the base claims and for any additional bacteria. This matrix just goes through the 3 different claim types, and then on the right side also indicates what type of testing is required for any additional bacteria that doesn't fall under one of the categories. So it's for the non-method bacteria, the additional claim, 2 batches of test subsets are needed to only test against 10 carriers per batch. So it's a reduced quantity of carriers, and you can even test the product concentration at closer to the nominal level. You don't have to focus on that lower certified limit as you do for the base claim. Also, the population required for the non-method species is quite a bit lower than the population required for the base claim higher level studies. And again, special considerations for the used solution. Each batch for Pseudomonas and Staphylococcus need to be tested on separate days to eliminate that day effect that can be seen. Alternate growth conditions in media can also be utilized for the non-method bacteria. Moving on to the 810-2300s, the sanitizers. There's a lot of different methods included in this one. There are some that are more frequently utilized, and those are the ones Talia and I are mostly going to cover. But some, these tables are read the same way as the previous ones. So non-food contact would be for use as a sanitizer, not a disinfectant, as a sanitizer in a laboratory space or an area that isn't expected to have contact with food surfaces. And then there's also methods for food areas that can be utilized. The one thing to take note of here is that for non-food contact for a sanitizer, for towelettes, there is no defined method currently, so that there is no sanitizer claim for a towelette. You would have to go for the disinfectant evaluation. The species challenge for each area differ, but the 3 batches are required, and the level of reduction is also different and with different time points that we will discuss as we go on.
So focusing in on that non-food food contact surface testing, it follows ASTM E1153. At times, you'll see sometimes laboratories or research facilities follow, depending on what they test or research, follow this method to validate cleaning their cleaning supplies used within the laboratory and choose an appropriate surface from there. The test surface is chosen depending on the proposed label claim. So oftentimes for hard, porous surface label claims, they are typically using unglazed ceramic tile as the sample carrier type. And for non-porous surface label claims, it's either like a stainless steel, a glass slide. I've also seen some types of plastic used as the surface. The organism is chosen based on that label claim. Of course, for this method, it's specifically Staphylococcus aureus and Enterobacteria. This test method is heavily quantitative. So to paint a general picture, the organism is typically introduced onto that surface. It will be set to dry, and then the disinfectant will be introduced, followed by a neutralization once that contact time is complete to ensure that the contact time is met. This mixture is then scraped from that surface and prepared in a sort of serial dilution and enumerated. This provides us with, in total, at the end, it will provide us with a % reduction, which can, again, can be used to compare different products, 1% reduction compared to another, the efficacy of that product to see if it met that contact time, of course, if it was appropriate for use. The huge benefit to this is that it can be used to evaluate very brief contact times down to the 15 seconds, which other methods don't allow us much flexibility for. Definitely, it's usually tested at 3 batches per test substance and 5 test carriers per batch, of course, with 3 control carriers. And the success of it will be a 99.9% reduction within 5 minutes when compared to mean recovery for control carriers. The next test method is for food contact surfaces. This is generally under AOAC International Chlorine and Disinfectants Germicidal Evaluation Equivalent Concentration. So AOAC 955.16. It's also a qualitative evaluation type of test. It's designed to identify the amount of test product that is equivalent to a known germicidal concentration of free chlorine. So to get a general picture for this test method as well, typically you'll set standard chlorine solutions at 200 ppm, at a known 100 ppm, and a known 500 parts per million concentration, and use it as a comparative scale against a test substance. So in a pattern of introducing a microorganism to typically a set standard of 200 ppm chlorine solution, and then transferring it and subculturing it and observing for growth, you'll continue a pattern of this subculturing into solution until you're able to, up to 10 times. From there, you'll be able to compare the values of the standards observed compared to the actual test, and evaluate success of this method. The focus organism are typically Salmonella and Staphylococcus aureus. There's usually 3 batches of test substance for this method. And its success is based on being able to demonstrate product concentrations equivalent to that of the standards that you prepared. These standards and subcultures are analyzed from growth, and essentially they must show a very similar growth of the microorganism to be successful.
So the next method is going to be the food contact surfaces, but this is going to be on the opposite end of the spectrum with non-halide active ingredients. So this one, we're using AOAC 960.09, the International Germicidal and Detergent Sanitizing Action of Disinfectants. This is a quantitative evaluation using water-soluble powders and liquids. It should be evaluated versus Escherichia coli and Staphylococcus aureus. Again, the ever common 3 batches of test substance independently prepared need to be tested. So this method is very similar to the ASTM E2783 in that it's a suspension test, when challenged with the product with a numbers control. So the success is a 99.999% reduction, so equivalent to a 5-log reduction within 30 seconds, so 30 seconds being the maximum contact time, when compared to the numbers control, which is prepared in a phosphate buffer dilution water or an SCI, depending on the needs of the test. So the next one is going to be again, a food contact evaluation, but this one is for towelettes. Towelette doesn't have a defined method. It does, however, have an archived webpage on the EPA that they use as a light guidance. So approval by the EPA for the protocol is definitely recommended. So this is for towelettes for a food contact surface, so for use in a kitchen. It is a quantitative evaluation using, again, for the food contact surfaces, E. coli and Staph aureus, 3 batches of the test substance. And then this is where it gets a little funky. 4 square feet of 2 different types of surfaces need to be evaluated using 1 towelette for all 4 feet. So one type of surface needs to be a hard non-porous surface, so stainless steel or glass surface, and one needs to be a cutting board with a rough surface, so a porous type surface. All testing needs to be in triplicate, and controls and carrier recoveries are required for this one. So to be deemed successful, we again need to see the 99.999% 5-log reduction in the number of each test microorganism within 30 seconds when compared to a control towelette. The kicker on this one is the control towelette is the exact same as the test substance with only the active ingredient removed. So we'll still have all the vehicles and surfactants in it, just with no active ingredient.
The next guideline we will quickly touch upon is 810-2400, which is disinfectants and sanitizers for use on fabrics and textiles. A disinfectant you'd most commonly hear for this is either a laundry detergent or some type of pre-soak treatment. If it's labeled as a disinfecting pre-soak treatment, it will be followed by the use dilution method, which encompasses Staphylococcus aureus, Salmonella enterica, and Pseudomonas aeruginosa, which are organisms typically found in nature or sometimes on human skin. If it's labeled as a sanitizing pre-soak treatment, it will follow ASTM E1153, which is will be modified to include 5% OSL. These organisms are Staphylococcus aureus, K. pneumoniae, or previous known as K. aerogenes. So to just emphasize the difference between these two, as mentioned, a sanitizer wouldn't be something considered broad spectrum because it's just intended for killing of bacteria. So that's why the disinfectant, if it's labeled as a disinfectant, it has to follow a different type of test method with, of course, the broad spectrum of organisms. If it's a disinfectant laundry, you usually use a simulated use study against S. aureus and K. pneumoniae. And again, these are other listed types of disinfectants under this category. What I want to emphasize in this case is that these are very much less developed guidances. It's no particular or rigid method for these disinfectants and sanitizers. It's very much a niche department of study at the moment, and it's still very much in development. So these are general guidelines that you can follow for a disinfectant or sanitizer that you're developing. But this is what's recommended for those for fabrics and textiles.
So in keeping with the niche market in testing are the last three OCSPP guidelines, and then including subdivision G. So air sanitizers, disinfectants, and sanitizers for use in water, products with prion-related claims in subdivision G, all require very specific test areas and methods, and entire rooms or certain size workspaces to make these all standard. And so the air sanitizers require an entire room or a box of appropriate size that can be monitored and growth collected. Disinfectants and sanitizers for use in water often require simulated in-use systems. So you would need the water pump systems and all that to see. Prion-related claims obviously require extra precautions to ensure no cross-infection. Subdivision G is a light guidance on testing of mold mildews. And so that requires special rooms with humidity controls to get the mildew growing, and then application of the test products have to come with a very special sprayer that's listed in subdivision G. As I said, these are very niche markets, and they require additional setups or laboratories to test. So interpreting the data is often the most challenging and one of the questions I get asked the most. The qualitative evaluation, so the used dilution or the germicidal sprays, you use carrier recoveries to prove that you went into the evaluation and ended the evaluation with appropriate recoveries from your carriers, but they do, as they're qualitative, they do not provide exact percent reductions. So these percentages can be inferred and estimated based upon your carrier recoveries and the fact there's no growth, but they cannot be used to substantiate label claims. The percentages cannot. There's also the quantitative evaluations, which use a control solution. So the ASTM E1153 uses a control solution to determine the percent and log reduction produced by the test substance. So this is where you use the control solution and expose it for the same conditions as the test substance to make sure that the reduction is seen from the product, not from the exposure conditions. Carrier recoveries are often done for these quantitative evaluations as well, but they are mostly used to verify population and not used for calculations. The big major caveat that comes with this testing, and one of the smaller loopholes the EPA has allowed, is if you're outside the ranges, either high or low for the population, there is certain caveats where a retest is not necessary. So using the first one as an example there, the mean control carrier count level is above the acceptable range and the test substance failed. So the EPA says, yes, a log retest is required as the test product was over-challenged by the microorganism. On the converse side, we have the next one down, the mean control carrier level is above the acceptable range, but the test substance passed. The retest is not necessary as the product was over-challenged but overperformed and still produced the reduction necessary. So they allow for all of that with even specifics for if there's contamination, and that depends on how much is contaminated, how pervasive is the contamination. Is it a laboratory failure? Is it a product failure? It just depends on the conditions if the retest is necessary there. If there's a documented control error failure, as in your sterilities end up being positive, the retest is required. And so they have different requirements for retesting, and the study director will have to justify those retests.
So we've been through so much information. I'm sure there's still so many questions because we've literally only scratched the surface of the amount of information and detail and specificity that this type of testing really needs. So the best recommendation we can give to you is to truly rely on that EPA website and those guidelines that are provided. EPA also does a great job of providing standard operating procedures for these guidelines, which are completely free and available to download. Even then, they offer additional guidelines for clarification on those SOPs. Definitely their website is a best practice to use, especially since these products will be submitted to them. If any modification is needed for your test method, of course, it needs to be discussed with them prior to you even performing the test. I've also listed a link for you guys to see those most frequently asked questions and see if you can find answers to your questions there, as well as the email for the EPA staff, who is always readily available to answer questions that you may have. If you have any other further questions about testing, you can always also reach out to Grady and myself. We are always happy and willing to assist.
Thank you, Grady and Thalia, for your insights. We've all learned some great information today, and you've given us a lot to think about. So I'd like to thank our presenters, Grady and Thalia, and we'd like to thank you, the viewer, for attending this session, and we hope that you found it to be a valuable experience.